Good morning to Dr. Shirley. I am Poya Hui, and for this assignment, I will be presenting on the title Gut Microbiome Dysbiosis, the Hidden Driver of Type 2 Diabetes mellitus. These are the main focus of today's presentation. Let's start with an introduction to gut microbiome. The gut microbiome community refers to the dense and diverse population of microorganisms that live in our gastrointestinal tract. These include bacteria, archaea, fungi, and even viruses. Interestingly, the composition isn't the same for everyone. It changes over time and is influenced by factors like age, gender, diet, and even geographical location. If we break down the bacterial composition, the majority belongs to four main phyla: Firmicutes, which make up most of the gut bacteria, followed by Bacteroidetes, Actinobacteria, and lastly, Proteobacteria. In this context, the first two phyla dominate and are often regarded as an important indicator for gut microbiota health. Next, a balanced gut microbiota is crucial for maintaining health. They promote food digestion, extract nutrients and energy from food, and even produce beneficial compounds such as short chain fatty acids. Moreover, they also regulate host metabolism, maintain energy homeostasis, and modulate our immune system. However, when microbiome community balance is disturbed, we face a problem which is known as dysbiosis. Dysbiosis can reduce microbiome diversity and abundance of microbiomes, disrupt microbiome metabolites production, weaken intestinal barrier integrity, and trigger systemic immune dysfunction. So, how does dysbiosis actually lead to type 2 diabetes? First, compositional shift can be observed. There will be an increase in Firmicutes, Prevotella, and Enterobacteriaceae, reduction in Bacteroidetes, Akkermansia, and Faecalibacterium. This increased Firmicutes to Bacteroidetes ratio, which is linked to insulin resistance. Second, there are functional shifts. Many of the pathways, including glucose and amino acid metabolism, will be reduced. The microbiome skew towards sugar and amino acid synthesis. This will lead to hyperglycemia, insulin resistance, inflammation, and oxidative stress resistance, which are the hallmarks of type 2 diabetes mellitus. Other than that, this biosis also leads to dysregulation of microbe-related metabolites. Short-chain fatty acid production such as butyrate, acetate, and propionate are reduced in type 2 diabetes mellitus. This usually improves glucose tolerance and gut health. Next, bile acids produced by gut microbiomes help regulate cholesterol levels and improve glucose tolerance and insulin sensitivity. However, in this biosis, their signaling to stimulate the receptor is disrupted. Third, branched chain amino acids will accumulate in plasma as there is a decrease in bacteroides and firmicutes that will help regulate amino acid metabolism. All of this will disrupt gut barrier integrity, increase intestinal permeability, allowing endotoxins like lipopolysaccharide to leak into the bloodstream, causing systemic endotoxemia and chronic inflammation. Endotoxemia happened when there was an increased adherence of intestinal E. coli and a decrease in bifidobacterium species. Endotoxins such as lipopolysaccharide directly damage the intestinal barrier, attract immune cells, and trigger the release of inflammatory cytokines such as TNF alpha, interleukin 6, and C reactive protein. These inflammatory molecules not only damage pancreatic beta cells, impairing insulin secretion, but they also contribute to insulin resistance in peripheral tissues. In short, this biosis links the gut environment to both the onset and progression of type 2 diabetes. Now, to study and even manipulate the gut microbiome, scientists are using gene editing tools like CRISPR-Cas. CRISPR-Cas is a natural immune system in bacteria that has been adapted for defense of the host against invading genetic elements. It works by using a guide RNA to target specific DNA sequences of the invaders, and a Cas9 protein to cut and modify the genome. Its working principles are as follows. First, acquisition. The system spots foreign DNA next to a PAM site, Cas1, Cas2 cut and capture it. Then, it stores a little piece, which is the spacer, into the CRISPR array. Next, CRISPR locus is transcribed into pre-cRNA, which is then processed into mature guide RNAs. Each guide RNA has a memory of one foreign sequence. Lastly, interference involves the guide RNA pairs with Cas proteins, find matching DNA in future invaders, and the Cas nuclease cut it, destroying the trait. With CRISPR, researchers can knock out gene function by cutting out a DNA fragment or insert desired genetic sequence into the genome by providing a reference DNA template. For example, Frolova and colleagues engineer human gut bacteria to enhance the production of short-chain fatty acids that improve both gut environment and metabolic health. Another study by Wang and colleagues engineered a butyrate producing probiotic Bacillus subtilis. This shifted the gut microbiome composition, improved glucose and lipid metabolism. These findings suggested that engineered probiotics could one day serve as a targeted therapy for metabolic disease like type 2 diabetes mellitus. To conclude, the gut microbiome is a key regulator of our metabolic health. These biases disrupt microbial balance, weakens intestinal barrier, promote inflammation, and ultimately contribute to the development of type 2 diabetes mellitus. But with advanced tools like CRISPR-Cas, we are moving closer to a personalized microbiome-based therapies. These are the list references that I use in this presentation, and thank you for listening.